Hello dear students, today is our final lecture and today our lecture will be about the genetic diseases with non-traditional inheritance. So, diseases with non-traditional inheritance, what it mean? It means that the diseases with non-traditional inheritance do not follow classical Mendelian laws. Or in another words, these diseases sometimes called non-Mendelian inheritance. Non-Mendelian inheritance in any pattern of inheritance in which traits do not segregate in accordance with Mendelian laws. These laws describe the inheritance of traits linked to single genes on chromosomes in the nucleus. In Mendelian inheritance, each parent contributes one or two possible alleles for a trait. If the genotypes of both parents in a genetic cross are known, Mendelian laws can be used to determine the distribution of phenotypes expected for the population of offspring. There are several situations in which the proportions of phenotypes observed in the progeny do not match the predicted values. So, what types of the disease? What types of the disease are the diseases with non-traditional inheritance? Look at the slide and you can see the different groups of this disease, and we will talk about all of this disease. The first group is mitochondrial diseases. The second group is the diseases of the trinucleotide repeats with anticipation. Next group, diseases of genomic imprinting, and the last group, hereditary diseases of somatic cells. So at first, let's talk about mitochondrial diseases. Mitochondrial diseases or mitochondrial disorders may be caused by mutations in mitochondrial DNA or in nuclear genes that code for mitochondrial components. As you know, the DNA in the cell are located in the nucleus, and also DNA are located in the organelles which called mitochondria. So the most genetic diseases will be caused by the mutations in the nuclear DNA, but in the same time, some diseases may be caused by mutations in the mitochondrial DNA or in the nuclear genes that code for the mitochondrial components. So, mitochondrial diseases have some inheritance features. The disease affects both men and women, but affected men can't transmit those diseases to their child. The next features is phenomenon of heteroplasmy, and the last threshold effect. What does it mean? So, the diseases affect both men and women, but affected men can't transmit those diseases to their child. But why? Why affected men can't transmit their disease? Because let's remember the process of fertilization, the process of making of the zygote. So when the oocyte connect with the sperm, only head of the sperm penetrate into the egg or in the oocyte and take part in the embryo formation. So the mite or connecting piece of the sperm does not penetrate to the egg. So all father's chromosomes stay outside from the zygote. So I remind you the structure of the chromosomes of the sperm. So sperm have the head, the mid, and the tail. And all mitochondria of the sperm are located only in the mid or connected piece. And this piece is not penetrate to the egg in the process of fertilization. So if the man have a mutation in the mitochondrial DNA, it can't be penetrate into the embryo. So that's why the disease affect both men and women, but affected men can't transmit the disease to their child. Let's see. On the slide, you can see the family tree with some mitochondrial disease, and how you can see this type of the genetic tree is the same like autosomal dominance type of the inheritance, but how you can see affected men have all healthy child, and you must pay attention when you read the family tree. Please pay attention for these features. The next features of the mitochondrial diseases or mitochondrial inheritance is the effects of heteroplasmy. Heteroplasmy is the lack of mechanism for the exact distribution of mitochondria between individual cells. So, during cell division, the mitochondria segregate randomly between the two new cells. Please look at the slide. When the cell divides, the pathological mutant mitochondrion divide into the daughter cells randomly. So in the one cell there are only one mitochondria with mutation DNA, but in the another daughter cell there are many type of the mitochondria with mutation DNA, and this is effect of heteroplasmy. Those mitochondria make more copies, normally reaching 500 mitochondria per cell. As mitochondrial DNA is copied, when mitochondria proliferate, they can accumulate random mutation, for example like this, with a high-level mutation. If only a few of the mitochondrial DNA copies inherited from the mother are defective, mitochondrial division may cause most of the defective copies to end up in just one of the new mitochondria. For more detailed inheritance patterns, we will talk about the human mitochondrial genetics some later. And mitochondrial disease may become clinically apparent once the number of affected mitochondria reaches a certain level. This phenomenon is called threshold expression, and it is the last features of the mitochondrial disorders, threshold effects. The tissue which have a low threshold of the mutant mitochondrial DNA are brain, heart, skeletal muscles, retina, kidneys, and endocrine glands. And the cells of this tissue are most metabolically active energy dependent, so in the mitochondrial disorder these organs will be affected frequently. So what disorders are mitochondrial? You must know this type of the diseases at first. Leber's hereditary optic neuropathy, Meles syndrome, or another word of the syndrome, torn muscle fiber syndrome, Lee syndrome, mitochondrial myopathy, and diabetes mellitus and deafness. Let's talk some about the clinical picture of these diseases. Leber's hereditary optic neuropathy includes some symptoms like visual loss beginning in young adulthood, because the nervus opticus is dead and the nervous fibers are in necrosis. And the eye disorder characterized by progressive loss of central vision due to degeneration of the optic nerves and retina. The next syndrome is Meles syndrome, or in another word, mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. Is one of the family of mitochondrial cytopathies, which also include MRF and Leber's, as we know, Leber's hereditary optic neuropathy. Meles is a condition that affects many of the body system, particularly the brain and nervous system, and also muscles. In most cases, the signs and symptoms of this disorder appear in the childhood following a period of normal development. Children with Meles syndrome often have normal early psychomotor development until the onset of symptoms between 2 and 10 years old. Though less common, infantile onset may occur and may present as failure to thrive, growth retardation, and progressive deafness. Onset in older children typically presents as recurrent attacks of a migraine-like headache, anorexia, vomiting, and seizures. Children with Meles are also frequently found to have a short stature. Most people with Meles have a buildup of lactic acidosis in their bodies. A condition called lactic acidosis, it means that the increased acidity in the blood can lead to vomiting, abdominal pain, extreme tiredness, or in another word, fatigue, muscle weakness, loss of bowel control, and difficulty breathing. Less commonly, people with Meles may experience involuntary muscle spasm or termen myoclonus, impaired muscle coordination, ataxia, hearing loss, heart and kidney problems, diabetes, epilepsy, and hormonal imbalances. And the next syndrome, now let's talk about the Lee syndrome and about its symptom complex. Lee syndrome, or in another word, subacute sclerosis encephalopathy. After normal development, the disease usually begins late in the first years of life, although onset may occur in adulthood. A rapid decline in function occurs and is marked by seizures, altered status of consciousness, dementia, ventilatory failure. and some words about mitochondrial myopathy. Mitochondrial myopathy are types of myopathy, as we know, associated with mitochondrial disease. On biopsy, the muscle tissue of patients with this disease usually demonstrate ragged red muscle fibers. These ragged red fibers contain mild accumulation of glycogen and natural lipids and may show an increased reactivity of succinate dehydrogenase and a decreased reactivity of cytochrome C oxidase. So, now let's talk about another group of the diseases with non-traditional inheritance. It is the diseases of trinucleotide repeats with anticipation. Trinucleotide repeat disorders are a set of genetic disorders caused by trinucleotide repeat expansion, a kind of mutation in which repeats of free nucleotides, or in another word, trinucleotide repeats, increase in copy numbers in regulatory untransmittable part of genome until they cross a threshold above which they become unstable. So this definition may look like tricky, but it is very simply. Let's see. Trinucleotide repeats. What it means? It means that in the DNA, in some part of the DNA, increase the number of trinucleotide repeats, or another word, the number of the triplets. Let's see in the definition. Increase in numbers in regulatory untransmittable part of genome. What it mean? This is difficult words, regulatory or transmittable part of the genome. It means in the exons, not in the introns, but in the exons. So exons, I remind you that it's a part of the gene with information about the primary structure of the protein. So this tricky definition of this type of disease, it means that in the some exons of the DNA increase the number of the triplets, and when this number cross some threshold, it causes different type of the disease. And anticipation, what it means? Anticipation is earlier manifestation and severity of the symptoms of hereditary disease in subsequent generations. For example, what it means? It means that father have a manifestation of the disease in the age of 30, for example, and have not seriously illness, but his son, his daughter will have more earlier progressive of the disease, for example, in the childhood, in the 5 or 6 years old, and have more seriously symptom complex, and this manifestation will be the example of the anticipation, is earlier manifestation in the childhood, for example, and severity of symptoms. The example of trinucleotide repeat disorders: Fragile X syndrome, Huntington disease, spinal and bulbar muscular atrophy, and spinocerebellar ataxia. And we will talk more about Fragile X syndrome and Huntington disease. Fragile X syndrome, or in the outer name, another name, Martin-Bell syndrome. The cause of the disease: increasing in copy numbers of trinucleotide CGG, more than 200 in the X chromosome. So, we said that the cause of the disease of trinucleotide repeats it's the increasing of numbers of trinucleotides or triplets, and these triplets can be different. For example, for Fragile X chromosome, it's triplet CGG, more than 200, some threshold for this disease, and in the exons of X chromosome. Physical features of Fragile X syndrome may include a long and narrow face, large ears, flexible fingers, and large testicles. About a third of those affected have features of autism, such as problem with social interactions and delayed speech. Hyperactivity is common, and seizures occur in about 10%. Males are usually more affected than females. This disorder and were finding in the X chromosome. So, in other words, Fragile X chromosome has an X-linked dominant inheritance. It is typically caused by an expansion of the CGG triplet repeats within the FMRN1 Fragile X mental retardation gene of the X chromosome. This results in silencing or mutilation of this part of the gene and the deficiency of the result protein, which is required for the normal development of connections between neurons. So, there are some tasks for you. Let's look on the slide, and here is two friends, Matthew and Benjamin. It's a real life, and one of them had the syndrome of Fragile X chromosome. So, on the basis of the symptom complex of this disease, please think who has the syndrome of Fragile X, Matthew or Benjamin? Think some 1 minute, and after that I give you a right answer. You can pause our lecture, think some 1 or 2 minutes, read one more time the symptom complex of the Martin-Bell syndrome, and decide who, Matthew or Benjamin, have this syndrome. One of these guys have a syndrome, and another guy without the syndrome, and another guy is healthy. So, and now I give you a right answer. The syndrome of X chromosome has Matthew, this guy. And now let's talk about one more diseases with non-traditional inheritance, the diseases of trinucleotide repeats, it's Huntington disease. Huntington disease, or chorea, is an inherited disorder caused by increasing copy of CAG more than 36 in part of the gene that results in the death of brain cells. The main symptoms are jerky, random, and uncontrollable movements called chorea. So pay attention, we said that in the fragile X chromosomes increase the number of triplets CGG, and in the Huntington disease the triplets CAG, another type of the triplets, and the threshold is different. There are 36, but in the fragile X syndrome more than 200. Huntington disease is a neurodegenerative disease. The earliest symptoms are often subtle problems with mood or mental abilities. A general lack of coordination and an unsteady gait often follow. As the disease advances, uncoordinated, involuntary body movements known as chorea become more apparent. Physical abilities gradually worsen until coordinated movement becomes difficult and the person is unable to talk. Mental abilities generally decline into dementia. The specific symptoms vary somewhat between people. Symptoms usually begin between 30 and 50 years of age but can't start at any age. The disease may develop earlier in life in each successive generation. And the next group of the disease with non-traditional inheritance are diseases of genomic imprinting. Genomic imprinting is the phenomenon when parents pass to their child completely identical genes, but these genes carry a specific imprint of the parent's gender. It means that the paternal and maternal genes are activated or repressed in different ways. Maybe now you think that this definition is difficult, but some later we will understand this mechanism and it will be more simply. So, genomic imprinting is epigenetic phenomenon. Non-morphological rearrangements of genetic material are inherited, but genetic activity we said in the definition that the activated or repressed function of the gene. This phenomenon is due to telomere methylation during gametogenesis. So, let's see. There are two different diseases, Prader-Willi syndrome and Angelman syndrome. The cause of this disease it is the absence of chromosomes 50 in the locus in the large large shoulder of the chromosome in the region 11 to 30. And the Angelman syndrome is the same, absence of copy of the chromosomes 50 in the same region. But there are two different diseases, the Prader-Willi syndrome and Angelman syndrome. And what the differences between these two diseases? The differences it from who, father or mother, the child gets this genetic disease. And if this mutation of the chromosomes inherited from father, the result of the disease will be Prader-Willi syndrome. But if the mutations are gets from the maternal copy of the chromosome, it will be Angelman syndrome. So the changes of the genetic material is the same, but it is very important from whom the child gets this disease. So let's see, this two type of the disease is very different, and we can see it in the different symptom complexes of this disease. For example, Prader-Willi syndrome is a genetic disorder caused by a loss of function of specific genes on chromosome 15, how we said some early. In newborn, symptoms include weak muscle, poor feeding, and slow development. Beginning in childhood, those affected become constantly hungry, which often leads to obesity and type 2 diabetes. Mild to moderate intellectual impairment and behavioral problems are also typical of the disorder. Often affected individuals have a narrow forehead, small hands and feet, short height, light skin and hair. Most are unable to have children. And now let's see the symptom complex of the Angelman syndrome on Angelman disease. It's a genetic disorder too that mainly affects the nervous system. Symptoms include a small head and a specific facial appearance, severe intellectual disability, developmental disability, limited to no functional speech, balance and movement problems, seizures and sleep problems. Children usually have a happy personality and have a particular interest in water. The symptoms generally become noticeable by 1 year of age. By the base of this symptom, that the children usually have a happy personality, this disease also called happy puppet syndrome. And the there are some diseases of genomic imprinting with the paternal inheritance. Let's see, we said the Huntington chorea, also Alzheimer diseases, psoriasis. Psoriasis is a disease of the skin, and malignant schizophrenia. And some disease with maternal inheritance. Let's see, narcolepsy. Narcolepsy is a long-term neurological disorder that involves a decreased ability to regulate sleep-wake cycles. Symptoms often include periods of excessive daytime sleepiness and brief involuntary sleep episodes. Another example of diseases of genomic imprinting with maternal inheritance is epilepsy. Epilepsy is a group of neurological disorders characterized by recurrent epileptic seizures. Epileptic seizures are episodes that can vary from brief and nearly undetectable periods to long periods of vigorous shaking. These episodes can result in physical injuries, including occasionally broken bones. In epilepsy, seizures have a tendency to recur and, as a rule, have no immediate underlying cause. Isolated seizures that are provoked by a specific cause, such as poisoning, are not deemed to represent epilepsy. And the next very interesting disease called L-Face syndrome. I think that when I said L-Face syndrome, you think somewhat like that. But L-Face syndrome, it means the traditional knowing on the elf and it's look like that. There's type of the looking of the elf. So what is the elf face syndrome, or in another word, Williams syndrome, is a genetic disorder that affects many parts of the body. Facial features frequently include a broad forehead, short nose, and full cheeks. While mild to moderate intellectual disability with particular problems with visual spatial tasks, such as drawing is typical, verbal skills are generally relatively unaffected. Those affected often have an outgoing personality, interact readily with strangers, and appear happy. Problems with teeth, heart problems, especially supravalvular aortic stenosis, and periods of high blood calcium are common. And this is a real photo of the patient with L-face syndrome. And the last group of the disease with non-traditional inheritance is hereditary diseases of the somatic cells. This group of the disease are caused by chromosomal rearrangements in somatic cells. The frequency of the disease less than 2%, and the result of these changes are follow in oncogene activation, and how a result it is a retinoblastoma, sporadic cases of congenital malformations, very different type of the autoimmune diseases, and senescence processes. So today our lecture is over, and it was our final lecture in this educational period. So now we will meet with you only in the next year. So have a happy new year, have a rest, and see you in 2021. Goodbye.